Okay, so let's go over the setup. We've got our plant. This is Alodia densa. It is an aquatic plant that normally lives underwater in freshwater lakes and ponds and streams. We've got our test tubes set up and they're already labeled LE, LC, DE, and DC. And we've got our beakers that are also labeled appropriately as well. And our water. That's just distilled water that we'll use for our pH probe. Our lamp, we use that for stimulating photosynthesis, plus a few other tools here. And our scale, so we can measure the mass of our Alodia. And we're ready. Alright, first thing we need to do is measure the mass of the Alodia and make sure that they're about the same. So I'm just going to take one out and pat it dry a little bit. And I need to make sure that my scale is zeroed. And looks like it is let's go with 1.2 grams. 1.2 grams for that piece. Okay, and then we'll see if the other one's close to that. Again, we want to pat it dry. And it looks like it's 1.4. So we're going to have to trim a little bit off. We want to make them as equal as we can to reduce any variability. It's not so much that we need to know their mass, it's that we want them to be the same. Okay, 1.3, not quite there. Okay, good. Now they're both at 1.2 grams. And we're going to place these in our experimental tubes. So the first tube is LE, light experimental, and the other is DE, dark experimental. I'll just poke them down in there. We'll use the forceps to get them down in there. Okay, and we want to add this water, but we can't add the water until we know what our pH is first. And before we use the pH probe, we need to zero it or calibrate it and make sure that in deionized water it gives a pH of 7 exactly. And I've calibrated it already, so you can see there it says 7.0, so our pH probe is ready to go. And we're going to measure the pH of our starting liquid. We can just do that in the big beaker since that's where the Alodia came from and that's the water we're going to use. And that's not tap water, that's the water that was in the tank where the Alodia was stored. Okay, and you can see there that it is 8.0. 8.0 is our starting pH. Now remember, this water came from the tank the Alodia was in, so it's not pure. It probably has some microorganisms and so forth in it. We need to fill these tubes with that water, and that'll be our starting conditions. Now, this is a control tube, so all it has in it is the water from this beaker. Our experimental tubes will have the plants and the water. Okay, we've got those pretty much filled to the top, just almost to the very top. And you can see that they are ready to go. The light tubes will stay on the desk. The dark tubes are going to be put away. We need to cover them up with something though, so I'm going to use this parafilm. Parafilm is really just a thin layer of wax that is kind of stretchy, and you just pull it across the top of a container to keep anything from getting in there. It's really mainly just to keep from any contamination while we're waiting for it. And you can see there it just kind of folds around and sticks, kind of like using saran wrap. So hopefully you can kind of figure out what the independent and dependent variables are for this. Should be pretty obvious. What are we manipulating, and then what are we measuring as a result of that manipulation? And as it said at the beginning of the video, it's real important for you to understand the relationship between carbon dioxide in water and how that affects pH. If you don't understand that, the rest of this won't make sense. Okay, we're ready to go. We need to put these in the appropriate location. So the first thing I'm going to have to do is cover up and hide my dark tubes, but I don't have any foil, so I'm going to use this black apron. And I'm going to wrap it up in the black apron. Normally we would use foil, but I just don't have any here. And I'm going to stick it in the cabinet where it's nice and dark, and that'll keep it from doing any photosynthesis for a while. Okay, the other tubes are going to go in the light, and it's not real sunny today, or else I just put them outside, but instead we're just going to use this light, and I want to try to get the light on it as bright as I can, and we're going to let them sit there and soak some rays. Okay, and now we just need to wait. It'll take about an hour for us to get some results. Okay, so as you can see, I've kind of rearranged where the tubes are a little bit so that they got as much light as possible. You can see that they're getting lots of direct light from the lamp, and what we're going to do is take the water out of these tubes and put them in the beaker, and we'll do the same with the tubes in the dark. They're down here in the cabinet, so I'm going to pull those out, and they've been sitting in the dark for an hour. And all we really need to do is transfer the water from the tubes to the beakers so that we can measure their pH. Okay, so our first one is our light control, so we just need to pour that into the LC beaker. And the light experimental will go in the LE beaker. And since we didn't really do anything to harm these plants, we can just pull them right out, stick them back in the beaker there, and we can recycle them, put them right back in the tank where they were. They're hardy little plants that do just fine. When you put them back in the water, they'll send out roots and keep on growing. Okay, our dark experimental. Again, we just need to pour the water into the beaker first. Make sure it's the right one. We'll dump that in. Okay, and then we can put that plant back. And finally our dark control. Okay, we're done with those. And now we just need to find out how being in the light or being in the dark affected their pH. And again, I want to make sure my pH probe is calibrated, and looks like it is. And then we're going to get our readings. Now as I go through these, I'm going to put on the screen a table that shows what the readings are, because to be honest, some of the readings were a little wonky and the pH probe wasn't acting right. I think maybe the battery was getting low. So use the data that you see on the screen, and you can pause the video if you need to see the initial and the final pH, and then use the data tables in your lab manual to go ahead and do the analysis. You'll have to account for the changes. And as you're doing that, notice that our final pH in the control tubes did change. And one of the questions would be, why would it change? Is there anything in that water that may have caused it to change? And I want you to think about that as you're doing the analysis here. And since the pH went up, what does that tell you? Does that mean that there's photosynthesis, respiration, one or the other? Is there another factor involved? What might be affecting the pH in the control tubes? And then once you account for that change, then how can you explain the final pH of our experimental tubes? What's going on there? Is it photosynthesis, cellular respiration, one or the other, or both? Alright, that's it. We've done our experiment. Now you just need to figure out what those numbers tell you.